Okay, let's talk about a huge challenge. By 2050, there are going to be nearly 10 billion people on this planet, and feeding everyone, especially with our climate changing, is one of the biggest things we've got to figure out. We need new, better, tougher crops. The thing is, the way we've been breeding plants for centuries is just, well, it's stuck in the slow lane. And frankly, we're running out of time.
So what if I told you we could take a process that usually takes, a decade of super careful work and just shrink it down to a single step? Yeah, imagine taking 10 years of waiting and trial and error and compressing it all into one. It sounds a little like sci-fi, I know, but it's a real biotechnology that's completely changing the game for plant breeding.
And that's where this amazing analogy comes in. Scientists actually call it the high-speed rail of crop breeding. It's this revolutionary shortcut that just blows past all the slow, scenic routes of the old methods. So how in the world does this express train to better crops actually work?
Well, first, let's really understand the bottleneck. To get why this high-speed rail is such a big deal, you have to see why the old way is so slow. And it all boils down to one fundamental challenge in genetics, getting to what they call purity. See, when you cross two parent plants, maybe one has great drought tolerance, the other has a really high yield, that first generation is a total genetic jumble. It's heterozygous. Think of it like a shuffled deck of cards. Sure, the traits you want are in there somewhere, but so is a whole lot of other stuff you don't want. To get a stable, pure line where every single plant has those winning traits, breeders have to self-pollinate and pick the best ones over and over and over again. We're talking up to 10 generations. It's a massive investment of time and resources that, honestly, we just can't afford anymore.
And this is where our high-speed rail pulls into the station. The technology is called culture, and it gives us an incredible shortcut to that all-important genetic stability.
So anther culture is a type of tissue culture. The goal is to take the anthers from a flower bud, that's the part that makes pollen, and basically trick those tiny individual pollen grains into growing into a whole plant. Now, because pollen is a gamete, a reproductive cell, it only has half the normal number of chromosomes, and that creates this really unique kind of plant called a haploid.
But a haploid plant on its own can't reproduce. So here's the really clever part. Scientists treat these little plantlets with a special chemical that causes them to double their own chromosomes. And the result? It's called a doubled haploid, or DH plant. And here's the magic trick. Because it was made from a single set of chromosomes that was just perfectly copied, the plant is 100% homozygous. It is genetically pure. That decade-long mission to get a stable, pure line? Done. Just like that, in one single generation.
Now, you might be thinking this all sounds super new and futuristic, but the science has actually been around for decades. I mean, the first haploid rice plant was made way back in 1968, and by the mid-70s, China had already used anther culture to release a commercial rice variety. So yeah, this technology, it's got a proven track record.
Okay, so let's pull back the curtain a little bit. Let's see exactly how scientists managed to turn a single grain of pollen into a fully grown plant right there in the lab.
The whole thing is this delicate dance of biology and lab technique. It starts with picking flower buds at the absolute perfect moment of development. Then, in the lab, they sterilize the anthers and place them on a nutrient-rich gel. This encourages the pollen to form something called a callus. It's this little undifferentiated mass of cells. From there, that callus is coaxed into becoming a tiny haploid plantlet, which then gets the chromosome doubling treatment to create our final, stable, ready-to-go plant.
And check this out. This image just shows the whole journey so perfectly. You can literally see it go from those tiny little anthers to this lumpy callus growing in a petri dish, and then finally to these vibrant green plantlets that are ready to be put in soil. It's an entire plant's life cycle just radically sped up in a lab.
It looks pretty straightforward when you lay it out like that, but believe me, getting it right is incredibly tricky. So what are the secret ingredients? What makes the difference between this working perfectly and failing completely?
Well, success really hangs on getting a few key things just right. And the most important one, by a long shot, is the plant's own genetics, its genotype. Some varieties are just naturally better at this than others. Timing is also critical. You have to catch the pollen at a very specific stage in its development. And of course, the parent plant needs to be healthy. But there's one more factor, a kind of secret ingredient that's, well, it's a little surprising.
That secret ingredient is stress. Yeah, you heard me right. To trick a pollen grain, which is genetically programmed to do one job and one job only, into becoming an embryo, you have to shock it out of its normal routine. The most common way to do this is with a cold pre-treatment. You're basically putting the flower buds in a fridge for about a week. That jolt of cold stress is the trigger that reprograms it, telling it to forget its original job and start down the path to becoming a brand new plant.
But even if you have the perfect technique, the perfect timing, the perfect amount of stress, there's still a major hurdle that has really limited anther culture for decades. And it all comes back to that number one factor, genotype.
Just take a look at rice, which is the world's most important food crop. You've got two main subspecies, Japonica and Indica. And as you can see from this chart, it's a night and day difference. Japonica varieties, they respond really well to anther culture. You can get success rates above 10%. But Indica varieties, they're notoriously difficult. You're looking at rates below 5% and a lot of the time way, way lower than that.
And here's why that is such a massive problem. Indica is the type of rice that feeds over half the world's population. It's the rice grown all across tropical Asia. So the very variety that stands to gain the most from this incredible technology has been the most resistant to it. Overcoming this resistance, this recalcitrance, has been the holy grail for rice breeders for a long, long time.
But that wall of resistance, it's finally starting to crumble. New breakthroughs are cracking the code, unlocking this technology for the crops that need it most. It's paving the way for a supercharged future in plant breeding. So think about it. What happens when you pair the incredible speed of an anther culture with the unbelievable precision of modern gene editing? Well, you get a system for creating better crops that's almost unimaginably fast and powerful.
And the technology making this possible is, you guessed it, CRISPR. You can think of it as a molecular scalpel that lets scientists make incredibly precise edits to a plant's DNA.
The teamwork here is just incredible, a total game changer. Scientists can use CRISPR to make a specific genetic edit, like say adding a gene for salt tolerance into an indica rice plant. But then, instead of spending the next 10 years trying to stabilize that new trait, they just use anther culture to lock it in instantly. A new, salt-tolerant, genetically pure line is ready in a tiny fraction of the time. This doesn't just speed up breeding, it speeds up discovery itself.
And this brings us right back to the ultimate goal. Anther culture is the high-speed rail that radically cuts down the breeding timeline. When you pair it with the precision of a molecular scalpel like CRISPR, you create a toolkit powerful enough to develop the resilient, high-yield crops we desperately need for our future. It's all about getting from an idea in the lab to a plant in the field faster than we ever thought possible.
So, we now have this incredible speed and control over the genetics of the plants that feed our entire world. The final question really isn't about the science anymore, but about us. As we master this ability to engineer the crops of tomorrow, how do we make sure these powerful tools are used wisely and fairly to build a food-secure future for everyone, everywhere?